All right, I think it's 1 minute past, so hopefully everybody's here. First of all, I'd just like to thank everybody for joining us today at this first OHDSI APAC Symposium Australia Chapter breakout session. We've got a great lineup for you guys this afternoon, and I really hope it helps you understand a bit more about data quality and the importance of data quality in doing network studies and working with observational data sets. So I'm just going to start now by sharing my slides, give you a quick overview of the Australian chapter, where we've come from, and a little bit about where we're going. Let's see. Okay, can everybody see that? Yep. I think, have I got it on the right view, or is that?
No.
No.
It's your view, yeah.
Not quite sure how to fix that.
You right click, you can, oh, sorry.
Perhaps if sorry, bear with me. Now it's opening up on my other screen. Let me just. No, I'm sorry, I'm not sure how to do that, so we'll just go with this. Okay, so OHDSI Australia. We first applied to be a chapter in December of last year, and we had our first chapter meeting on the 9th of December of that same year. So as you can see, we're a very new chapter, and as you will have seen, Mui explained to you a bit about the other chapters, and some of them have been around a little bit longer than us, and we hope to follow in their footsteps and really sort of activate observational research in our country. We now have 84 members, and we have created a website, ohdsi-australia.org. So if anybody online is not already a member and would like to be a member, please jump on that website, and there's a section for registering as a member. So go ahead and do that. Even if you're not from Australia, we're quite happy to have you as a member. And here are the institutions that are involved in sort of the running of OHDSI Australia. So UNSW, Tang and Jitendra, who are both presenting today, University of South Australia, so me and Ty, and University of Melbourne, so Dougie and Roger Ward, so Dougie also presenting today. So again, other institutions, we're very welcome to have you. So Mui's already been through this, but I just wanted to highlight that as part of OHDSI Australia, we are trying to develop a webinar series. So far, we've had 4 separate webinars. We're always inviting international collaborators to give us that sort of global perspective. So we were lucky enough to have Nigel Hughes, who talked to us about the Eden network. And Eden really is, I think, a gold standard of how we might sort of work towards becoming a collaboration here in Australia. There's seminars, there's training opportunities, and there's lots and lots of research going on in that network. So check out eden.eu, and there's a ton of things that you can look at, training opportunities and things to learn a bit more about OHDSI and OMOP. We had Mui also talk to us just recently. That was great perspective of how you actually translate data and talking about data standardization. So we'll be having another series of webinars next year. So some of our activities, we've been really getting on with the mapping of data sets here in Australia. Lots of primary care EMR databases going on in New South Wales and Victoria, and a large database which contains over 700 general practices from all over Australia is being converted at the moment, led by Tang. Some secondary care EMRs, hospital EMRs. mainly based on the Cerner system at this stage, but hoping to expand that to other systems as they come along. So quite a few across Australia. And of course, the administrative claims databases are being translated at the moment as well. So in terms of network collaboration, we've been participating widely in lots of the different community events. So we've participated in the study-a-thon, the symposium. As Mary said, we're looking at getting involved in the research study that was presented yesterday on second-line hypertension treatments. And that really stems from the work, at least I was involved in the LEGEND study, which was the first study on hypertension treatments. We did a workshop for the International Population Data Linkage Network Conference. So that was really great, just introducing the data linkage community to the CDM and how you might translate your data into the CDM and what would the benefits be for that. So that was really exciting to be part of that conference. And some research grant proposal, which I'm pleased to say we've actually had some success in funding this research here in Australia. That's my next slide here. So we've actually got a few different research grants awarded just recently. One by Tang to do that translation of that primary care data set. Another ARDC grant led by Dougie to do some hospital EMR translations. And I was lucky enough to get a Centre of Research Excellence in Medicines Intelligence, which also will help to translate some of those administrative claims databases here in Australia. So I'm very proud of the efforts of our community, and hopefully that will fund more and more work in the area. Also wanted to shout out to our poster presenters. So we'll have a short break at the end of this session, a breakout session, and our poster presenters will be there online on this channel right now. And if you have any questions, please feel free to ask them and get involved or write them an email. A lot of the work that we've been doing for OHDSI Australia has really been umbrellaed by the ARA collaboration, so the Australian Health Research Alliance. And really the goal of that is to help accelerate the pace and the scale of research translation. So under that umbrella, we've created the Transformational Data Collaboration, and really getting together some of the key people across Australia to help uplift the data sets here in Australia. So we have three sort of streams in the ARA Transformational Data Collaboration. The first, which we're going to be really getting into today, is the data quality assessment stream. So that's really talking about how we can improve the quality and the applicability of routinely collected health data. Another work streams around terminology and terminology mappings. Really important here in Australia that we have our own community who are helping to map our vocabularies into the OMOP vocabularies, and of course the use of the common data model is a stream that I'm leading in that group. Dougie's going to talk a bit more about the priorities at the end of the session, but at this stage, our focus has been on this data quality and interoperability, and today's session is all about that. We'll talk about the White Bandicoot tool, and if people don't already know, a bandicoot is kind of like an Australian rabbit, and you will know that there's kind of a lot of rabbit references in OMOP, so white rabbit, rabbit in the hat, Usagi, and so on. So at the end of all of this, I hope we can promote some of the conversions of data sets in Australia, and so that we can participate in those network studies and really activate our community, not just for phenotyping, for everything. So this is our website, odyssey-australia.org. Jump on and join the journey with us. So I'm going to now hand over to Tang, who will chair the next session. So thanks, Tang.
Thanks, Nicole, and thanks and welcome everybody to the next session, which is on data quality assessment. Let me just share my screen.
Do I have to maybe stop sharing? Right, sorry, go ahead, Tang.
Oh, no, wrong one. Can you see that now?
No.
Oh, maybe I have to get out and share again.
Yep, we've got it now.
Got it now?
Yep.
I'm going to put it into presentation mode. Okay. Welcome. Can you see the slides? Yep. Welcome, everybody. Before I start, it is the custom in Australia to acknowledge the traditional custodians of the land on which we meet. So I'd like to do that now, acknowledge the traditional custodians, as well as acknowledge all Indigenous elders, past and present. My name is Tang Liao. I'm a clinical informatician with an interest in optimizing the use of real-world data contained in electronic health records to produce real-world evidence that are fit for purpose, as kind of outlined by Patrick in his opening session yesterday. The quality of evidence is obviously very important. However, as EBM practitioners or evidence-based medicine practitioners, real-world evidence needs to be contextualized to patients and practitioners. So this panel session will be framed within the user-centered principle and the quality of the evidence, and therefore the quality of the data. So the basic question is data quality assessment of data sets and data networks. So there'll be multiple networks, which is what OHDSI is all about. The basic question is, does the CDM add value to data quality assessment? But I guess it's more open than that, and we hope participants will have lots of questions, and hopefully the panel will address them adequately. So the focus of the panel is essentially on examination of the data quality assessment and management in both CDM, right, and environment, as well as the non-CDM enabled environment, and presentations by Claire and Dougie. So the first two sessions I will provide a very broad framework, and Michael Khan will provide the data quality framework, and this will frame the discussions by Dougie and Claire on the White Bandicoot and the OHDSI DQ data quality dashboard, respectively. And then Jitendra will try and bring it together by describing very briefly the project that we just been funded by the Australian NHMRC to do, which is looking at comparing the CDM and non-CDM enabled data quality assessment. So we need to look at data quality from the data life cycle perspective. From left to the right of the diagram, and I'll very briefly just highlight that. From data creation and collection into primary databases, and then extracted ETL into secondary databases. Hopefully, as time goes on, we don't have to do the ETL at this level, but at the primary level, but that's another day and then another time. Why? I guess before that, I just to put and I forgot to mention that the main reason why we're doing this comparison between CDM and non-CDM environment is that, as you know, as Nicole has pointed out, Australia, and indeed like the rest of the world, many of our data networks are not mapped to a common data model, but hopefully a lot of them are contemplating now, and hopefully we'll get to moving towards the home of CDM. So we need consistent data quality assessment in both environments during this transition. Now, in terms of the data quality life cycle, it goes right through from there, right from left to right. Data collection and data creation, extraction, and hopefully use. So the first important thing is governance. We need to think about. And that includes governance at the local level as well as the secondary data warehouse level. Second thing we need is provenance. And that's important to be able to trace the origins, and the authorities are the custodians under the responsible for the data sets that's being brought together. And the governments need to consider compliance to the FAIR principles, which are findability, accessibility, reusability, and interoperability, as well as the 5 safes in terms of privacy and security, which includes safe projects, safe people, safe data, safe settings, and safe outputs. And the framework for data quality, and you'll be familiar with Professor Khan refuses to let me refer to this as the Khan framework. So I'll call it the Harmonized Intrinsic Data Quality Framework. So, and most of you will know that it covers conformance, completeness, and plausibility. And both Michael and Claire and Dougie will go through it in details. But we also did a bit of work with Michael and the Australian Institute of Health and Welfare on reviewing the literature to see what we need to do in terms of assessing some of our local data sets. And that includes the data from the My Health record. And we can talk about that later, but this is a national personally controlled health record system. And we ended up adding a contextual and a technical category. But this is probably secondary to the what we're going to discuss today, but it's important. And these are, sort of represented here in yellow. But actually, trying to represent this into this contextual and technical categories may be done through the CDM, or may be done through the common data model, a common method data model. And there's a bit of thinking going around. And if we have time, we might have a bit of chat with it at the end of the session. But finally, to introduce this session, we need to think about that data quality is an important driver and indicator of digital health maturity, because that's what we're all about. Kind of, how do we harness digital health to improve healthcare and health? And health informatics is a information-rich discipline and is driven by data. So in the digital health maturity framework that we use, we have 4 essential digital health foundations looking at the ICT infrastructure, the essential digital tools, readiness to share data, which is coming very relevant to what we'll be talking about today, and health system adoption. So if you look at the right here, as we go up from the maturity from a basic level, which is an unpredictable system, a control system, still getting predictable, but still inconsistent, standardized, which is stable, consistent, predictable, and addressing requests from users. And then optimize, where the focus is on improvement and improving services, and then innovation at the end of the day. But data is very important. Data interoperability, sharing, and data quality. So just giving that very, very big picture. So with that, I'll come to the panel. I'll summarize a bit of their biographies from the online program. So, sorry. With this kind of broad background, we brought this group together to help us look at data quality assessment and management and discuss the data quality in both CDM and non-CDM enabled environments. We'll start with Professor Khan, who will discuss data quality assessment, principles and practice. After his talk, he will hand over to Claire, who will discuss CDM enabled data quality assessment using the dashboard as an example, or SD's example. And then followed by, who hand over to Dagi, who will talk about the White Bandicoot as an example of a non-CDM enabled data quality assessment. After which he'll hand over to Jitendra, who will describe the study that we funded to do to compare CDM and non-CDM enabled data quality assessment. And then we will get into the panel discussion. So please post your save your questions or post it on the chat, and we'll look at it and answer it. But we'd like to leave the question and answer session to the end, along with the discussion. So without further ado, I will hand over to Professor Khan. Over to you, Michael.
Michael, you're muted.
How about now? Does that work?
Yes, that's great. Thanks.
Very good. And hopefully you should be seeing my screen. Let's get
Your
to
very
my
cute dog.
Yes.
Yeah. Yeah, she runs the house. She's 14 years old and manages the whole house. Okay, are you seeing my screen?
Yes. Oh, great. Thanks.
Are you seeing it full screen rather than in presenter mode?
Yes, full screen.
Yeah.
Perfect. Okay. All righty. Well, thank you. Thank you for the opportunity to speak with at the conference. I appreciate the opportunity. So I am Michael Kahn. I'm a emeritus professor at the University of Colorado at Anschutz Medical Campus. And the work that I'm presenting is work that has been done at the Colorado Center for Personalized Medicine, which is the location where we bring together clinical and genomic data to develop models for personalized care. I run the Health Data Compass data warehouse, which brings together both clinical and genomic and epidemiological data together to support those activities. And the work that I'm going to be presenting really was done sort of in the context of supporting that project. So this is a figure that I stole from Dr. Lau in terms of the same article that he showed his diagram for the digital health maturity levels. This is a different figure from the same article. And stealing from the upper left-hand side of his diagram, I went ahead and sort of said, expanded the perspective of what we want to talk about in terms of data maturity and especially data quality, to expand outside just a health system's maturity level, but really think also more broadly. And that's part of the reason why I believe that most of you are here at this conference, to understand more broadly how we can take the data that we all have and share in both, and not only for our own health systems that we represent, but regionally, obviously nationally for the Australian context. And OHDSI has always had, you know, a very international perspective. So broadening our perspective to go beyond just our own institutions, while that's important, and data quality in the context of a single institution is important, we also want to keep our eye on regional, national, and international data quality challenges. So, you know, any conference or any talk on data quality has to start with a definition. This is a very standard one, comes from Joe Jurin, and I'll just read it to you. Data are of high quality if they are fit for their intended uses in operations, decision-making, and planning. Data are fit for use if they're free of defects and possess desired features. Now, this is a very industrially oriented definition. Joe Jurin was an industrial engineer, and so it has a kind of a lot of that kind of flavor to it. But the key point that is relevant to what we're talking about is the underlined intended use. That data quality is really linked to its use, and that makes it very context sensitive. And some examples are that, it is quite possible to do very interesting data discovery using data that have flaws. You do not need, perfect data to do science. And the other sort of corollary to this idea of intended use is that the very same data set may have very different data quality features depending on how you want to use it. And a data set that might be outstanding in one context may be deficient in another context, and vice versa. So context is something that this community that we struggle with, and that we really need to sort of better understand. And you'll see this idea of context and fit for use keep coming back with the other slides. So one thing was mentioned was the harmonized data quality model that you'll see appearing in others' talks. This was motivated by really my consternation in trying to understand what was going on in the data quality literature, and in particular in the biomedical data science literature. And what you see here are 10 different publications across the top here, including Dr. Leal and myself, contributing to what is somewhat of a cacophony of terms being used to describe, similar or different aspects of data quality. And it was very difficult to take a publication, take two publications, and figure out whether or not they were talking about the same thing, or were they measuring the same thing, or was this different, or how was this different? Just a real headache in trying to get through the literature. And so I was fortunate enough to obtain a grant to bring together a cast of like-minded individuals. You can see their names here. And again, Dr. Leal's right here in the middle of the thick of things, as he always is. And really bring together a community of people together and try to harmonize the way we talk about data quality. It's a terminology harmonization. It doesn't harmonize the measures, but trying to have the right kind of places to put a particular data quality idea, so that as we move this community forward, we're at least hopefully putting roughly the same things in the same place in the same terms. And I'm just going to go through the harmonized terminology just very quickly. This is not a lecture on the harmonized terminology. It is available. This document here is an open access document, so it is available to you. But it divides the data quality world into two major contexts: verification and validation. And verification is that very first level of data quality assessment that one typically does using your own data and taking a look at your own metrics and sort of saying, do I look okay relative to what I expect for my organization? It's very much a single organizational perspective. Whereas validation steps outside of your world, looks for gold standards, looks for other sources of comparator data in a network environment, may look at other sites as comparators, and really sort of says, where do I fit relative to what I think are either peers or gold standards? So there's these two views of the world, one more inward, one more outward, verification, validation. And then at the highest level, there are these three categories, of some of which have subcategories underneath them, which I won't go into. But at the highest level is different ways of thinking about completeness, which is really just asking the question, are there any values there at all? Forget whether or not they're good or bad. Are they even there? The next level down, which is called fidelity, is, do they line up the way I expect them to line up? Are they, somewhat related in the way that I expect them? A lot of that has to do with time. Time is an important dimension in healthcare. And then plausibility, which is basically, are the data believable? Are they accurate? Are they what I think do they represent the idea or the concept that I think they should? So, we're talking about data quality practice. They want to make sure that this session was very practice oriented. And so this diagram really kind of lays out where in sort of the data lifecycle data quality practices occur. And I've hinted at it previously in the framework. There's these phases, one dealing with what does my data look like at the site level. If I share my data with others, what does my data look like in a multi-site level? And then we talked about this fitness for use idea. What does my data look like in the context of doing a particular scientific study? You see a lot of cycles because data quality assessment in general may discover anomalies. You go back and you revisit your data, you make changes. This happens both at the site level and at the network level, and hopefully that doesn't happen at the analytic level. For us, and what we're going to focus here is really on these two phases of data quality assessment, the site level and the multi-site level. There's a lot less work that has been done in the literature on study-specific fitness for use measures. It is the next area that this community needs to start to address, and Claire has done some work. I don't think it's being presented this time in creating a tool that will help a little bit on number 3. We're going to focus on 1 and 2. So we're talking a lot here. I work mostly in health records as opposed to administrative databases. And so my focus has been mostly in looking at where data quality issues arise in the context of using clinical records. Looking at sort of the cycle of taking data from the time that it's entered in the context of a clinical care event, and then it's stored in some sort of database management system that manages and may back up and sort of takes care of those data. Then at the time of one wants to do a research study, those data are extracted out either into a CDM or to an analytic data set. One does analytic computations on that and then displays and reports them. If one just qualitatively takes a look and says, where does data quality issue, where do they kind of arise from? This is my own sort of, unscientific, highly biased opinion that really the vast majority of data quality issues arrive at the time of data entry. These are very busy people who are giving clinical care. They're not here to sort of enter data for our research studies. And so anything that can be done quick and easy is done in these environments. Yes, we can make mistakes and sort of have programming errors in our ETL code that does the extraction and or apply incorrect measures or have analytic programming bugs or even sort of either misleading or incorrect displays. Far less likely in the place in which data quality occurs. Really sort of entry is the leading bugaboo in this area. And why is this the case? Sort of why is it that electronic health records? Well, these tools, as I've mentioned, are used by very busy people whose focus is on delivering clinical care and not really on data capture. And so these tools, all of them have been optimized for workflow efficiency above anything else. And so a lot of templates are used, a lot of copy and paste, a lot of shortcuts go into the data collection at this phase. And really, these systems are purposely made to have very minimal data validation checks because these get in the way. And yes, you may have min and max limits or perhaps drop down and pick lists and let you know something is or is not required. But this is about it. There's very little other kind of data validation because, again, these would stop workflow. So I have some examples of some of the fun things some see in the health record. I'm sure you have your own ones that you've kept in your drawer, and I'd love to see some of the more interesting ones that you find in your examination of health record data. But here's an example. This comes from Nicole Wyckoff at Oregon Health Sciences, and what she did is took a look at the distribution of systolic blood pressure. So this is systolic blood pressure against the counts for a particular value for systolic blood pressure. And in her examination, one sees, this kind of distribution kind of looks like a porcupine kind of figure. I'll stop just for a moment just to let you think a little bit about why this may have happened, kind of what the leading source of this might have been. Hang in there for a second. I wanted to see where I am on time. Okay. The etiology of this is that these were done at the time in which blood pressures were manually recorded off a manometer, which is a column of mercury, and human beings have a tendency to want to round values to the nearest 5. So each of these spikes is either 5 or a 0 on a continuous scale. It'll be interesting to see now that we've mostly converted to electronic measurement of blood pressure. I'm going to repeat this study in the last year or so, where all of our clinics have now converted to electronic blood pressure measurement to see if, and hopefully these biases aren't there. Here's a similar example. This comes from the Mini Sentinel project from Jeff Brown. This is a project where they were extracting data. This was the 7th cycle of extracting data. This is a value that is rising over time, as it should. The JAGs are fine. On the 8th extraction, all of a sudden, there's out of the clear blue sky comes this enormous loss of data in an old period of time. So they're doing an 8th extraction. They're out over here. They're doing a historical extraction, and suddenly there's this huge divot in the data. Jeff was really never able to explain to me where this came from. I do have another example where this was explained, something very similar. This is another a site where this is the previous extraction, ETL cycle number 2. All of a sudden, same code, same data set, a quarter later, ETL cycle number 3, and it looks completely different. It has, again, this very spiky view, and it has a completely different baseline. The baseline now is up here compared to there. Out of the clear blue sky, 1 quarter later. This one was explainable. This one came from, first of all, the baseline was the acquisition of a new organization that was merged into this organization. And this spike was a change in the business rules, where the business office said, on December 31st, we're going to close all visits that the physicians have failed to close, because we can't bill for visits until they're closed. And so on December 31st, under this new rule, they went back and they closed off all the end of the year visits, and that's where this came from. Taking a look at the other environment, where we're now looking at a multi-site situation, I only have one example here, and this comes out of the HMORN network. This is a large network of health systems across the United States. This is a study that was looking at patients who were getting agents that cause you to retain potassium. Your body holds on to potassium, and that puts you at risk for having elevated potassium levels if your body holds on to the potassium for too much. So hyperkalemia is a life-threatening complication of the use of these agents. Here are 4 different study sites, and something is clearly different with site D relative to everybody else, both in terms of their cohort counts. They're getting half of their cohort percentages than everybody else, and they're not seeing the nature of the outcome that all the other sites were seeing. And this is something that becomes very obvious when you do a multi-site study and you lay sites up next to each other. Now, one of the things that we were interested in finding out is, people have been measuring data quality for quite a long time, and there's a number of different publications out there that describe various data quality programs. So I had a student, Tiffany Callahan, reach out to 6 different data networks. This is about 3 or 4 years ago that Tiffany did this, and these are very different networks. OHDSI is included here. And she was able to acquire from those 6 networks roughly 11,000 different data quality checks that were done. And she used the Harmonized framework, which again was developed specifically so that we could put like things together, like kind of measures with like kind of labels, and compared kind of where were people spending their time in these 6 networks doing data quality. And the leading place, let me just check my timing. Okay, the leading place in which data quality was being done was in plausibility. Do the data make sense? Atemporally, not looking at them over time, but these are mins, max, means, 25, 75% quartiles, these kinds of very simple statistics. More interesting is this diagram from her study. This takes each of the 6 networks and says, well, where was this network spending its time? The other slide had put everybody together. And the only thing to take away from this slide is the distribution of the colors is very different across these 6 networks. They are spending their time in different ways in different parts of the data quality landscape, if you want to use that word. We're not saying who is right or who is wrong. What we're just saying here is that different networks have made different decisions in terms of where they want to invest their time in data quality. This comes from the PedsNet network. This is a pediatric network that we participate, and by far and away, the #1 leading cause of data quality in their area was missing data. Not surprising for all the reasons that we've mentioned about people who are very busy collecting the minimum amount of data necessary for either clinical care or billing, and they are just filling out what's necessary to move on to the next part of clinical care. Another interesting finding that came out of PedsNet is as they matured their data quality program from version 1.0 to version 2.0, and they released more and more sophisticated data quality checks, and they had progressively more rules and checks in there. The more they looked, the more they found, not surprisingly, the more data quality issues were uncovered as the program was able to dig deeper and deeper into the data. So now I'm coming to the end of my time here and the end of my talk. I'm queuing up the talk that you're about to hear from Claire and Dougie to sort of raise this unsolved issue that the current data quality methods, the current state of the art right now, is many of the tools about data quality, either the measurement computations or the visualizations that are associated with them, right now are tied to a specific data model for which they're written against. So whether or not you're writing R or SAS or Python or SQL, whatever you're writing here that is developing your measures. We talked about means and mins and max, but there's also temporal trends. There's counts over time. There are many other ways in which data quality measures are computed. The current methods, for the most part, are tied to the data model of which they're written against, and then the visualizations to take a look at them again are tied to data models. So this is an old slide. It doesn't include Claire's tool. It doesn't include Dougie's tool. That's going to come on next. But you can't take an Oscar computation and apply it to the mini Sentinel data model and display it with a Caesar dashboard of data quality. Caesar may have something you really love. They may have a really wonderful visualization, but if you're sitting over here in OMOP v5, you can't use their visualization because their visualization works only against their output format. And similarly across all the others, you're pretty kind of stuck into a data model, a computation against that data model, and a visualization against the output of that computation. And this is really a shame because there's innovation that's really occurring both in the computational side, especially with advanced now AI models, machine learning models coming out, really being able to detect and learn new anomalies. I'm working with a computer science PhD student who is working on automating data quality detection. And she's come up with incredible methodologies. It's tied to the OMOP data model. You can't do it if you're sitting over here on Mini Sentinel. And really my vision, my hope, my dream, my hope that Dougie's going to solve this by the time you get to the third lecture today is really to kind of change this idea and really come to a common way of structuring data quality measure output so that each of the computations, each of the tooling that people are creating will, instead of writing to a data model specific structure, writes to this common structure that is data model agnostic. It's just the measures, it's the data quality measures. And then we rewrite our visualizations against this common model. So it doesn't matter whether or not Caesar or Mini Sentinel or OMOP created this JSON. And it doesn't matter also to the visualizations, sort of where it came from. And that way, if someone does something really cool and, Claire's out there making the data quality dashboard, really hum and sing and just kind of, dance and everything, and you're sitting on Caesar, your Kaiser Permanente, your national health system, love to be able to use that. So that's not yet here. People are thinking about it. Hope to maybe that we can motivate this way of thinking. So this is my last slide. I'll wrap it up here. It's really important, I think most of you who sort of practice with data management, to really appreciate and make sure that your leadership appreciates. It's really a message for leadership that local data quality work and network data quality work is a large effort. And it really is a marathon. It isn't something that you do and you say, check that box, we've done data quality, we can go on and now do something else. It's really is a marathon that you just need to continually invest in. You need to continually staff, and leadership needs to understand that. But what do you get back from it? Why do it? What's the ROI on a strong data quality program? Well, it really is higher quality research, and more than that, it's really trust and transparency of what are the data and how strong are the data that underlines the science and the evidence that comes out of all of our work. So I'll end there. That's the last slide. We've asked that people hold their questions to the end, so please write them down. This is how you can reach me if you have other questions that you'd like to ask. I'm going to stop, if I can figure out how to stop owning this, or maybe Claire, you could take over somehow.
I think I can. Let's see. Are you seeing my screen?
Yep.
All right, perfect. Awesome. Well, thank you so much, Dr. Khan. I really appreciate your talk. And I think I have so many things I could say in response to it, but I only have a short amount of time, so I won't go too far down that road. But what I'm going to be sharing with you guys today, as he alluded to, is really this CDM-enabled data quality tool that we've developed in the OHDSI community. And it's not quite singing and dancing yet, but we're hoping that we can get it there. So let me just a few slides. All right, so I'm just going to take you quickly through our solution right now for investigating data quality. And I think it's helpful just to talk quickly through the structure of the tool and the rules that are applied. And some of you may have seen these slides before, so I apologize in advance, but I think it's helpful to just get an understanding of how this works and why the structure of the CDM is important for it to work. Oh, Jitendra, I think you started sharing.
We lost your slides, Claire. Yeah.
Yeah.
Sorry.
Is that better? That's okay, no problem. So then I'm also going to take you quickly through a couple examples of the tool in action if I have time. I have a demo to share with you. And I'm just going to talk through a couple of the networks, not just OHDSI, but a couple of OHDSI, other OHDSI networks, I guess, or networks that use the OMOP Common Data Model would be a better phrasing, and how they're using it, and how we might be able to get to that state that Dr. Khan and Dr. Liao are talking about, where it would perhaps this tool could help bridge that gap to allowing us to have some sort of CDM agnostic data quality. But of course, that's really going to be Dougie's area of focus. All right, so we took that harmonized framework for data quality that Dr. Khan was talking through as we sat down to build this tool, and we weren't quite sure where to begin and really what data quality checks we wanted to include. If you saw that paper he was talking about by Tiffany Callahan from 2017, and you might have noticed that at that point OHDSI had the fewest number of checks across those partners and only had 172 checks they were reporting. And we kind of wanted to start from scratch. And so we sat here and we said, you know, how do we fill out this table to like actually start thinking about data quality in terms of this harmonized framework? So the first thing we did is actually we tried to define what a data quality check actually is. What is it that we're going to use to measure data quality with this tool we want to build? So we decided on this definition here is that it's an aggregated summary statistic that can be computed from the data, to which we can apply a decision threshold that determines whether or not this statistic meets expectation or whether it passes or fails. And I'm really going to just focus on that highlighted part, highlighted in yellow. I'm not going to get too far into the thresholds today just for timeliness, but we do have a lot of information on our website about how this works if you're interested. So kind of going off of that definition, we said, okay, let's just define one data quality check we're interested in. And this is one that comes up often, and I think many of us start with, which is how many people in my database have a funky year of birth? Or, you know, in this case, how many people in my database have a year of birth in the person table that occurs prior to 1850? And it occurs 1850 because it seems like so crazy that if someone has a birth year prior to that, it's probably an error, like that entry error that Dr. Khan was talking about. And so what we did is, as we wrote some of these data quality checks, and we wrote them in this specific way, is that we started to find that these things like year of birth, person, 1850, these items, these values are not really specific to the actual statistic that is being calculated. So they're just things we can potentially swap in and out, but the underlying math of that data quality check doesn't really change. So what we ended up doing as part of this tool, and what I think is kind of unique to this tool, is that we've abstracted the idea of data quality checks into something more generic, which we've coined just a data quality check type. Not a very inventive name, but it's something that we can use as this like abstracted data quality idea. So where our specific check of the year of birth now becomes the number and percent of records with a value in some field in a CDM table that's less than a low value. And by abstracting it, what we can start to do is stop in different CDM tables and fields and values. And so this is why the structure and use of the CDM is important here, because we know what tables and fields to expect in a database. So we can then know what we can swap in and out to these data quality check types. And so if we apply this to a different example, thinking about days supply in the drug exposure table, instead of year of birth, we're now looking for drugs that have a day's supply less than 0, because most likely that is also some sort of error, and we'd want to address those records in some way. And so we're using the same statistic, the same calculation as the year of birth calculation, but now we're looking for wonky days supply values. And so what we did in our first version of the tool, there's a couple different examples of some data quality check types. We've defined 20 of them so far, and many of them are related to the CDM itself. This past year in the Common Data Model Working Group, what we did was we actually worked to bring together the specification of the model itself with these checks, so that they really do work together. But what that means is that this tool is very focused on conformance to the model itself. And you'll see that in some of these data quality checks. So is it a foreign key? So if you're familiar with the Common Data Model, you know we use specific standardized vocabularies that are based around concepts. So we check to make sure that every concept field in a table has its foreign key or has its value also in the concept table itself. But that is a very OMOP specific check. But it's important if you are working within the OMOP network or OHDSI network that we make sure that all your concept IDs actually exist and that they are standard and valid. So yes, it is CDM specific, but it is important also for OHDSI and OHDSI enabled network research. So we started with these 20 different check types. And when we actually defined all of these checks using this kind of tool that we've developed to swap them in and out and swap these values in and out, we ended up right now with about 3,351 different unique checks that can be run against a single OMOP CDM instance or database. And I was going to touch a little bit on thresholds, but I think I'm going to skip this in the interest of time. But just understand with the understanding that this tool does not tell you, it will say whether or not your data quality checks pass or fail, but it is all user defined. So it does not mask the actual final result. It gives you the final result and then tells you whether or not that final result passes or fails based on the definition that you have given it. We do have a set of default thresholds that come with the tool, but it is extremely flexible per site and per database. Yeah, so I'm gonna just skip this part in the interest of time. So the way this tool works is that, as I was saying, you define the checks you're gonna run in that list of 20 different check types. Then you feed it your thresholds for how the tool should determine whether or not your checks are passing or failing. And then it spits out this really nice little dashboard here that is totally contained in the R package. So you do not need anything really beyond R to be able to bring up this dashboard and take a look interactively at your results. All right, so I'm going to go through a couple different examples of the tool. First thing I'm going to do, let's see, I have 6 minutes left, I think. So let me quick bring up the tool just to share with you how this looks. So I chose a US Medicaid database, mostly because I was reading about the Australian healthcare system. Of course, you can tell I am American. And I think that this is closely related to the healthcare system that you guys enjoy, that lovely single payer healthcare. So this is a database that is comprised of state-funded administrative claims. And you can see it brings up a nice overview, and you can see our, not the con categories, as we've been told not to call them that, but the harmonized data quality categories and contexts, and how many checks that were run in each intersection of a context and category, and the number of passes and fails. And there's some metadata about the database. I'm hoping we're going to get a little bit more into this metadata common data model, which Dr. Liao teased up, and I'm actually very, very excited about that because I think it's sorely lacking. So this is what we have now, at least for this database in terms of metadata, but I'm hoping to hear more maybe from Dougie and Jitendra about that. And then it actually gets you into the next piece, gets you into the results of the tool. And this is really where this is helpful and interactive, and you can look through all the different checks that were run in different ways and filter them down. And a lot of this was born out of that Achilles tool that showed up in that paper, that Callahan paper from 2017. We still use it. It is still extremely valuable, but there were some things that were lacking, and we actually built them into this tool, knowing that we really needed it. So, for example, each row here represents a data quality check that was run on this data, and you can see there's a lot of passes, which I'm happy about. But one nice thing is if you expand it out, it gives you a lot more information about it, like the number of rows, the number of denominator rows, and then it also gives you the query that was run. So if you're not familiar with SQL, that's okay. But I'm sure whoever is in charge of monitoring the quality of your database would be very happy to see this, and we find it really, really useful. And there's just a couple other things. So there's the option to add notes, and you can filter if they do have any, you can filter that, and that will show up as you look through your different checks that were run. So don't want to go too far into that, but I just thought it's helpful to give kind of more tangible example of how this tool works. All right, so I've got a couple minutes left. I just want to touch on some of the networks that are currently using this tool and how they found it helpful. So we've got the European Health Data and Evidence Network. And if you've been around OHDSI, you've probably heard about them before, but they are born out of a large IMI grant that was given to Erasmus University, as well as there's like a public-private partnership, and it's managed by Janssen Research and Development, where I work. And this is basically, they are, I think, trying to do what you guys are doing now, which is they are trying to build a large network of European data that have all been converted to the OMOP Common Data Model. And that's a really large undertaking. So what they are doing is they are using the data quality dashboard to help assess a database's readiness for inclusion in network research. Because I'm sure, as you guys are finding now, it can be really hard to get up on all the different OHDSI tools and get your data in the CDM, and building out that infrastructure can be challenging. And so what they've done is use the DQD as a way to figure out which databases are ready to participate in research. And earlier this year, I shared this tool with the US Food and Drug Administration, and they have been using it as part of their BEST initiative, which is they are essentially looking to use observational data to answer some of their clinical questions around biologic safety and vaccine safety, which is, of course, a huge topic right now. And so some of their data partners have been using the data quality dashboard to help increase that trust and transparency that Dr. Khan was talking about in the data that they are using. And then as well, there's a US National Institutes of Health grant that has put together this National COVID Cohort Collaborative. And what they are doing is grabbing COVID patients from different EHR systems, really university health systems, and Dr. Khan might know a little bit more about that. But they are basically taking COVID patients from around the US and different university health systems and bringing them all together into one giant COVID database, and they are using the OMOP Common Data Model to do it, as well as they're using the DQD as part of their data quality, I think they call it their data quality pipeline. So they have a bunch of different things, and they are really interested, of course, in data quality at the source as well, which is, you know, where this tool doesn't really work right now. This is really once it's been converted to the CDM, but I think what you guys are going to be talking about, what Dougie and Jitendra is going to be talking about with White Bandicoot, is going to be thinking about how do we assess quality on a database that hasn't been converted to the CDM, because it's still, of course, incredibly important. And just thinking about how we might reach beyond the OMOP Common Data Model, which, of course, you know, this is the OHDSI Australia chapter, so we are interested in the OMOP Common Data Model, but I think we want to reach beyond it a little bit, because data quality should be agnostic of data model. So what I wanted to just share a little bit is how this tool could potentially get us moving in that direction. As I was talking to the other panelists the other night, I was thinking about how this tool works, and really, as I was saying, what you feed into the tool are these checks you want to run, as well as the thresholds that your checks will be evaluated against to determine whether or not they pass or fail. But really what this file is actually the schema of the database. So yes, right now it works on the OMOP CDM, but that's because I feed it in the OMOP CDM schema. I list out all the tables and fields, literally in a CSV file, like almost like an Excel file, where I just say, these are the tables, these are the fields, this is what you look for, and run these checks on these, you know, tables and fields. And so I really got to thinking, and one of my colleagues in Eden has actually started thinking about, what if we gave this tool a schema that didn't look like the OMOP Common Data Model? And it turns out it works. So, and so what I'm hoping is maybe, you know, we could start to think about it not just in terms of the CDM, but in terms of data quality across the board, and how do we get to that, you know, that one JSON file that would work with everything. And maybe we could start to think about it in these kind of terms, and how this tool kind of really sits on top of just a schema that you specify, and it doesn't have to be the OMOP CDM. But the way we would need to do that really is we would need some robust metadata and information about the schema, and we need to figure that out, because sometimes they can be very complicated and very convoluted. So I think with that, I'll hand it over to Dougie, and I think he's going to take it from there and take you through White Bandicoot, and maybe how we can get even closer to non-CDM data quality. So, thank you.
Thanks, Claire. Dougie.
Okay, thanks very much, Claire. Can you finish your sharing? I'll see if I can get mine to work. Okay. All right. Right screen. For some reason, I'm seeing every screen but the one I want.
Yes, is there?
That it? Okay, that's fine. Okay, so can you see, is that going full screen on the opening slide? I can't quite tell.
No, you're on slide 26.
That's not right then, just a moment. It's a bit weird. Screen. How's that? Is that showing the first one now?
Very good.
Yes.
Yeah.
Yep.
All right, technology. Goodness gracious, who would ever trust me with any of it? Never mind trying to write tools for data quality. Look, thanks very much to all the presenters so far. I mean, Michael, you seem to have some great aspirations that I'm going to solve all of our non-CDM quality problems. It's quite a challenge. So we're really at very early days with this. And it's fantastic to see where Claire's thinking is here as well. So I think it's very, very complementary. So I think it's really exciting. I sort of first came into data quality from a different angle, certainly not from the common data model angle. A couple of years ago, we did a survey to try and find out what primary care databases are available in Australia, and it's due to be released pretty imminently. And it was quite sobering. So this has been mainly my areas working in the primary care space, and in Australia we've got 106 different data sets. This was as of a couple of years ago. And of all of those, there was only one that was mapped to the OMOP Common Data Model, and that's TENGS. And a third of the data custodians, when we asked them, didn't use any form of data quality framework, and in fact an awful lot of them didn't really sort of understand what that meant. The quote here is sort of puts it into context. You know, what's important is that accessing primary care data for secondary purposes is a monumental waster of time. Data cleaning is high cost, low value. The quality means research conclusions are on shaky grounds. It's an exercise in futility. You know, so this is where we're starting from to a certain extent in Australia. The GP systems don't conform to any particular standards for data collection or anything. So they represent some of the hardest sorts of data to try and utilize for research because it needs so much work. And you know, I'm really keen that we can support better research and also validating that. And of course, a few years back, we got the record statement coming out, which builds on Strobe to try and give a checklist of items to try and in our research papers demonstrate the lengths we've gone to validate the quality of our data. You know, in terms of things like bias, how did we handle the variables? How did we handle missing data? And what are the sort of cleansing mechanisms that we've used on the data? To give an example, so here's this is one of our main Australian GP systems. Just zoomed in on a particular screenshot. And in this system, there's a couple of buttons that sit very close to each other. So you can click a button for the reason for a consultation, and you can also click on a procedure button. And when you do that, the screens are almost identical. And what actually happens is people quite often click the wrong button. And even within here, you know, you can still pick from the same lists. So although one's a reason and one's a procedure, you can get all sorts of funny things happening. So we see it. So this is actually data that's just put in as an example. And what we've actually got here is data in the underlying tables of the GP system that ultimately would end up getting used for research. And what we have is in a progress table, we've got the visit date recorded as the 21st of November 2018. And then we've got two diagnosis records, which are down as the 22nd of November, and the field's called diagnosis date. And the reason is nausea. So nausea is being recognized as both a diagnosis and a procedure. And when you actually look at what the GP sees on the screen, indeed, the record is associated with the 21st of November, not the 22nd. And it's got here procedure nausea. So what's actually happened is this diagnosis date at the top isn't a diagnosis date at all. It's actually the date that the information was entered that corresponds to the episode. Okay, so when I look at that, I just think, well, how can I deal with that? And I was in Australia. We've a couple of years ago, the Australian Health Research Alliance was formed with some national funding to try and bring together all in an open manner, researchers across the team, as well as 78% of all acute services as well. So there's real opportunity through this open collaborative networking capability to try and think, well, how can we change things gradually at a fundamental level? And my area, of course, I'm specifically thinking about how we can transform data and try to do things that are difficult to do in our normal competitive research arena. So I want to get beyond the competition. So White Bandicoot comes out of that from the Australian Health Research Alliance Transformational Data Collaboration. And we've heard a little bit already about why it's named that. And the principles of the tool are really to have an openly accessible tool that can assess data quality on any data set. Because I'm thinking about these 106 different GP data sets, never mind any other ones. We looked around for the best framework internationally to build it on, and we eventually did choose the Harmonized framework. And this was before, in fact, we were thinking about common data models. Certainly we were thinking more around the data frameworks. And what I wanted to make sure as well is that we were able to capture some of the qualitative information, some of the non-numeric sort of information that impacts quality. And the example I showed you previously it gives you some idea about how people can make mistakes in the original system when they're even collecting the data. And that's the sort of gem of knowledge that we'd love researchers to have when they're looking at their data. So we wanted this to work regardless of the intent to map to a common data model, but we also are big fans of common data models and OMOP in particular. So we're really interested in developing tools that can actually build and integrate. So what does this look like? Well, if you take an example, in Australia, you've got the GPs are just starting getting performance incentive payments around quality. So what happens is that we've got the GP EMR system over the left, and the government's coming up with the definitions for the indicators. The data elements are then identified, and ultimately data is extracted, and the statistics go back to government. But the question is, can we actually trust the figures that are going back to government? So who actually does the transformations, and what do they mean? And ultimately, you can actually have transformations happening at all sorts of different levels that impact quality as well. Every time you transform data, you're potentially making a mistake or impacting what the data looks like. So what we're seeing here is that ultimately, from a single EHR vendor system on the left, you might end up with a whole range of different data sets with different attributes and quality metrics. And ultimately, a researcher, what they will see from all of that is basically different data sets. And here there's an arrow going, for instance, to one that might be based on OMOP. And who's correct? Good question. So you'll be familiar with these sorts of tables. So Michael and Claire both talked about verification and validation. So we've been applying this to these quality indicators, in particular around things like diagnosis information, and also here reason for prescription. And we're really trying to build on some of the good work that's out there. So we're looking at the work that Claire's been doing and how the queries are being put together and what they mean. Now the challenge is that when you're not using a common data model, how do you define your queries? The way your data is structured can be infinitely variable. So we don't have a complete answer to that one, except for the fact that when you write the queries in White Bandicoot, you can apply them across multiple tables and things. So once you've got some of these written, they can be applied very quickly across a large number of tables and fields. So this is a first view of the White Bandicoot system. On the left is a part of the screen where you can select the database you want to connect to. It supports just Microsoft SQL Server at this point, but the idea is for this to be generically able to connect to anything that can work with a SQL syntax. So if I zoom in just a little bit, what's happened is we've connected to a couple of different databases, and the lower one here that's highlighted, what we've actually done is we've run two different analyses, if you like, for two different projects against Granite Project Snapshot. So this is just a database, and we've wanted to look at different sets of tables and fields from them. And when you actually start annotating the information about its quality, that information works across any different analysis that you might do of that particular data set. So over time, for different research projects, you might run this tool and look at different fields, but the information gets retained across them all and updated if people find out new issues regarding the quality of a particular system. So here what we're doing is looking at the data at the table level. So what's happened here is White Bandicoot has scanned the schemas and automatically populated all the table names. And what's happening here is a researcher then is going in and manually annotating the additional metadata and the table descriptions. But also we can record quite a lot of detail here. So these are flags. It's difficult to see here, but you can record flags and additional information to bring specific problems with the data or ways you need to interpret it to the attention of the researcher. And you can filter your tables, so you can go from very large schemas and narrow it down to exactly the things that you're needing for your research project. And here we're actually seeing an example of a SQL query. And as I mentioned earlier, we're actually using the same syntax and structure as Claire. So we're trying to build on the excellent work that is already happening and to try and make what we're doing in its structure compatible as much as we possibly can. And here we're seeing data inside a patient smoking table, and what you're seeing here is all of the queries replicated across, so these are completeness queries that are replicated across all of the fields in a table, and you can do that by pressing a button, and we can run individual queries or you can run them all. Okay, so once you've actually run the queries and annotated, how do we actually display that? Well, what we wanted to do is to come up with something that was web-based, because our idea here is if you've got lots of people collecting all of the quality metrics, as for instance, as Claire was showing earlier on, we would like to be able to have the data visualized in a standard tool. Now, this isn't terribly good. I think Claire's team are really the experts in this space, but we're just getting started, and I think we'd like to work closely with Claire and the team to see where we take this. So basically, within the tool, once you've uploaded your results, you can then visualize them. You can further narrow down the information you want in your report and then hit generate. And then it starts to show the data for each table. And you can see some of the metadata that's in here. So this one, patient smoking table, saying BP does not capture smoking assessment date. It's a separate field, and it's telling us other things. In the interest of time, I'll just move on quickly and finish this up. But we can see data graphically as well, looking at incompleteness across fields. And this is still under development, but we can also then look at tables and look at their percentage completeness, conformance, and plausibility. And we can choose to just look at the graphical output on its own here around each of the fields. So nearly finished. Sorry, I'm 5 minutes over. If we look at the aspirations then of White Bandicoot, what we've been trying to do is to develop a tool that allows us to run a data quality assessment across different databases. And I've got the fact that maybe we want to use someone else's tool, so I'm not necessarily trying to say that our tool is going to be the best one for doing that data quality assessment. But what we're interested in is, like Claire's dashboard here is an R. What we'd kind of like to do is make something that's web-based, and it means that anyone in the world, so long as they have permissions, can actually get in and visualize the quality metrics of any of these databases, whether they're OMOP or whether they're based on an entirely different schema. And what we're really looking at beyond that then is if you take a database and convert it to OMOP, what we'd like to do is add on the metadata that's actually held in the dashboard, so that you can click on from within OMOP, within the CDM data, you would actually have the additional metadata to click into this web tool. So here we are with OMOP with that extra metadata. So the idea is that the researcher can use that and gain access to the additional information in there. So fundamentally what that means is here's a sort of common slide about the tools that are available through the OHDSI collaborations. And what we're thinking is that White Bandicoot has a role here for databases that don't necessarily get converted to the common data model, but where they are, we're really interested in having the CDM able to link back into the output of White Bandicoot, as well as potential tools like Achilles. Thank you very much.
Thanks, Dougie.
I'll hand over now to Jiten.
Thanks, Dougie. Okay. Can you see my screen?
I can't see it yet, Jitendra.
How about now, Dan?
No.
Yep.
That 1200 baud modem just isn't working.
Do you want me to share from here?
Yeah, if you can, or maybe let me give it a try again one more time. I can see that the screen share is not picking up. Let's see. Let me close the slides and open it.
That's worked.
Yep. Okay, so can you see my screen, okay?
Yes.
Okay, all right, sorry. Thanks, Dougie, for the presentation. Also, thanks to Michael, Deng, and Nicole. I think it was really good background, especially in terms of why data quality is important, especially in the context of both CDM and non-CDM. So with this, what I want to do is I quickly want to provide a summary on new project we will be working starting from January 2021. For those who don't know me, I'm a research fellow at the School of Population Health based at UNSW Sydney, and also a member of WHO Collaborating Center for eHealth based at the school, which Teng is leading. So this is an HMRC partnership project which we applied back in 2018 for projects commencing in 2020. But I think due to the contracts, we actually requested an extension. So officially, this project will be starting from 2021 January. So we've got quite a long list of investigators. So we can see some familiar names who are very active in OHDSI community, including Dagi, Nicole, myself, and also Teng. So this project is, it has a really good number of partners, but essentially very focused on primary care. So NPS Medicine Insight or NPS Medicine Wise, these two are the programs run by the National Prescribing Service. So we are interested in, or actually should say, we want to evaluate some of the hypotheses we have using the Medicine Insight database, which is a very large primary care database, which includes about more than 700 general practitioners. And it's a very good national representative sample of GPs in Australia. And in addition to NPS, we have Central and Eastern Sydney PHN, and also we got a GP Synergy, and also we got local Western Sydney local health system. So the main objectives of this particular project is actually we want to do data quality assessment before converting into CDM and after conversion of CDM, and it's not just about the data quality assessment. What we want to do is we wanted to do the data quality assessment to test some of the hypotheses using multiple use cases we actually identified. So one of the hypotheses we have is, we got about 3 hypotheses. So the first hypothesis is the data quality as we measure using the standardized framework or harmonized framework. We believe the data quality will be improved after converting into CDM. And the second one is the data quality of specific CDM-based queries is improved. Again, it's very similar to the hypothesis 1, but the idea here is that CDM-based queries improve the data quality when compared to non-CDM-based queries. I think the rationale behind here is that because once you have a structured CDM queries, you tend to have more agreed standards, and also you have more documentation around what is what. So the chances of introducing any new errors, or like how Michael mentioned, whether the errors could be analytical or even for simple SQL queries. So I think that's the rationale here for hypothesis 2. But this is the most interesting hypothesis, at least for me. I think some of the aspects have been nicely covered by Teng and also Michael. One of the key things is actually the data management and the technical cost efficiencies which are associated with the OMOP CDM. We don't know much. I mean, so I think it's one of the very important criteria to consider as well before converting into OMOP CDM. So that's another hypothesis. What we're looking at is we'll be able to manage the data efficiently once we convert into OMOP CDM. So again, maybe in the slide, I just want to expand a little bit further, like why we wanted to do particularly data quality assessment and why this particular project. I just want to emphasize that the routinely collected electronic health records are semantically very different from administrative claims data. At least in my opinion, I feel they're very complex and they're more very granular. They're very heterogeneous. Another important thing is they tend to capture complex clinical pathways, which are often very simplified in administrative claims data. So to give a very good example, let's say, for example, I'm interested in looking at chemotherapy regimens for IPMN of cancer, intraductal papillary mucinous neoplasm of pancreatic cancer. So usually we can get that information only from electronic health records, because you don't have that granularity of staging and also the morphology required for a given specific disease. But often in the claims data set, you would only find it as pancreatic cancer. So I guess so that's the complexities and the granularity we're dealing with electronic health records. So it really depends on the sort of research questions you're asking. So as I said, so it captures very complex clinical pathways, and the other often slightly, especially in Australia, at least based on our research experience, what we realized is that electronic health records are very good for to understand and investigate the localized health service utilization, and also for clinical audits as well. We can use administrative claims data and other data sets, but the problem is, as I said again, we lose some sort of granular information. Again, to give a very good example, let's say, for example, we have a hospital with multiple outpatient clinics, let's say psychiatric clinics and also child psychology clinics, and I want to see the pathway between these two clinics. So usually this level of granularity is missing in claims data, because often they tend to get aggregated and reported and coded, of course. So there's a little bit of loss of information. So that's the key rationale behind why we wanted to do this, especially this particular project. So in addition to the ones I mentioned earlier, it also introduced very unique challenges, especially when we wanted to convert into harmonizing EHRs. So let's say a very good example would be ETL conventions play a very, very important role. A very good example is about the provider ID. So I think in the OMOP common CDM, we can only have one provider ID, and the convention they suggest is to just take the last visit provider ID. But if you actually follow some of these conventions, we will not be able to answer some of the research questions. So, for example, a continuity of care index, which is one of the most widely used indices in primary care and also in the secondary care to understand the associations between outcomes and also the health service utilization. So this is a very good example. So when we actually harmonized EHRs, it also introduced a certain number of unique challenges. We believe that if we take a systematic approach, something like data quality assessment, or even looking at the data quality life cycle, what's happening to the data right from start to end, or some of the concepts even highlighted by Michael as well. So if we actually take a look at a proper systematic way at different steps, probably we would be able to at least identify and characterize these challenges. So instead of just, I think it's important we are aware of these challenges, because the evidence generated after harmonizing the CDM should be carefully considered as well. So I think that's one of the reason and also one of the rationale for this particular project. Another interesting one was, so far, as far as we are aware, there was no economic evaluation aspects discussed in terms of CDMs in general. Not just the OHDSI CDM as well. We actually had experience dealing with both I2B2 and Transmat CDMs in the past in 2014 and 15 for cancer data sets, and also omics data sets. But what we observed was we always expected there would be some sort of economic evaluation coming up, so we could use it as a guide, especially when you're scaling up to large data sets, and especially for government and government bodies, and also in academic settings. I think economic evaluation is very, very important. Sorry. So these are also some of the aspects we thought we could address all these research gaps using our project. So this is at a very high level. So we are calling it as a Sydney EPBRN. Basically, we got a nicely laid out governance and management. I don't want to get into the details of the exact the governance and management. So, but what I would like to bring your attention is to just look at these working groups. So we've got a cost evaluation working group, and we've got a data quality and interoperability working group. So these two are actually cross-cutting across the 4 use cases we identified. So this is what this project will be informing. So we have, at the end of this project, the outcomes will be informing, what are the cost efficiencies, what are the technical efficiency, how would it pan out if you want to adapt CDM. So that's one aspect. And the other aspect is, as I highlighted in the previous slide, some of the hypotheses would be answered, whether the data quality will be actually improving, and whether some of the specific queries to run particular studies will be improved. So we got 4 different use cases. We actually selected these use cases based on experience, and also after a lot of brainstorming, because we had to select the use cases in such a way that they actually reflect some of the complexities. Maybe I don't want to get into too much details, but maybe we can discuss a little bit more around this in the data quality panel. But the gist is, like, we didn't select randomly these use cases. We selected very unique use cases in terms of primary care setting, and also in terms of CDM and data quality. So the process is, so we're going through this process currently. So we have a governance set up. So the idea is to develop individual study protocols for each use case. So I'll skip through the ethics approval. So basically, we want to get ethics approval. The final objective is actually, depending on the research question and the suitability of the research question, we might want to, or at least we want to consider running OHDSI network studies for some of the studies. Out of the 4 use cases we presented, we believe at least 2 or 3 studies could be really good OHDSI network studies. So I'll let you know, guys, once it's once we have some concrete protocol in place. So this is just the timeline. We're looking from January 2021 to all the way to end of 2023. Hopefully everything goes according to our plan. So that's it. I think if you have any questions, please feel free to contact either Tang or I.
Thank you, Jitendra. Well, I'm sorry we kind of about 12 minutes over time, but we have enough time for some questions from the audience. But while they're thinking about it, let me share my screen. I guess that's the fundamental question that we had. Was, does the CDM add value to data quality assessment and management? Will the CDM or the ETL process improve data quality? Can it? Kind of fairly broad questions, but I guess that's some of the uncertainties behind this proposed program, and also the discussions we've had. So I'll open it out for discussion by the panel first, while I'm waiting for questions to come in from the audience. So I think the issues that Michael raised about the challenge of data quality assessment tools that agnostic of data models, and perhaps leading to common data models and common metadata models, maybe. But it's an interesting area to work on. So perhaps I can throw this to the panel. Say, what would you expect to come out from this? From this panel, but also what do you expect to come out from the project that Jitendra just described? As he has indicated, we often lose data and especially due to the loss of granularity from the ETL process. And maybe I can throw it out to Michael first, and then the others chime in as we go along.
So I will sort of address your question, but there's also a great question from Karen in the meeting chat. So I want to make sure that we're sensitive that we have an audience question. I have to say, I'm going to answer your question in a way that I don't think is where you think it's going to go. The value that we have gotten by adopting a CDM and working towards a standardized pipeline has really been access to a community that is behind that CDM. And in particular, the OHDSI community is a pretty, dynamic and very vocal and very thoughtful community who's really worried about making sure that the CDM data supports good science. And having that community back us up in terms of our thinking about how do we how do we do a transformation? How do we move a data element? How do we think about a whole data domain? And really making sure that we're thinking about the analytic use case, which I think is going to get to Karen's question, sort of there is changes in data that are meaningful, and there are changes in data that are not meaningful for the analytic use cases. It's been really the community that has been far more valuable to us than the actual technology. It could have been really any common data model, but a community like the OHDSI community who keeps us honest about how are you going to use that analytically? Is there real meaning in that data element? Are you really sort of trying to harmonize, sort of something that is never, ever going to be used? That has been where the value has been. It's been far more in the community than in the technology. Probably not the answer that you wanted, but that's where we have found value. I'll stop there.
Claire, then Dougie.
Yeah, maybe who wants to go next? So you want to go, Claire?
Oh, no, you can go ahead, Dougie.
No, it's absolutely the case that any time you do conversions on data, there's room for mistakes. But also, I would say is that if you don't understand the data properly that you're going to convert from one format to another, I think you're in a world of pain there as well. And that's why I'm really interested in trying to look at the quality metrics and what the data means before, or at least as early as you can get in the stage through the different conversions. My thought is that quality can actually improve over time. You might not necessarily get everything correct the very first time you look at data because your understanding increases. But if you look at things like this conversion to the common data model, people are defining their whole ETL process. If you've made a mistake in that, it's possible to correct it over time. But you need to be continually looking and be able to annotate and collect more information about what the underlying data is to gradually increase that quality. So there's no simple answer there. We can get it wrong, but the only way to get it right, I think, is the sort of approach we're trying to do here.
Yeah. Claire?
Yeah. So I definitely agree with both Michael and Dougie. And I have to say this was my first introduction to the White Bandicoot tool, and I am very excited about it. I had to turn my video on. I don't know if you saw me, but I was like doing a little dance. Like I really, really love it. So I think you're doing great work. And I'm very excited to apply it to my own data, and I can't wait to get my hands on it. But to Karen's point, in terms of changing the underlying semantics or the underlying data, I think, as Dougie said, as long as you're continually evaluating your process, you're not going to change your underlying data. As long as you are the data expert, you know what's there, and you know what it should look like at the end, which I think is a big key in that. And there's a lot of testing that goes into that. I know my group, for context, at Janssen Research and Development, I manage anywhere between 10 and 13 different large databases that have been converted to the common data model, and they are data sets from around the world. And it is, there is a lot of testing that goes in to make sure that what you put in gives you what you expect out in terms of the CDM. But there's, I think, as long as you're looking at that, you're not changing the underlying data. And in my opinion, you're making it better because you're making it work for you. And then you do get that community behind you, as well as we make a lot of use of all the tools that are available within the OHDSI stack. And it's amazing what you can do with those things.
Can I just add that recently while we've been trying out the tool here, we actually found what we think is a major issue with what's happening with the national quality improvement indicators. So it was White Bandicoot that found this. So in fact, sometimes the tools can actually uncover things because you're running so many metrics across your data set. So what we've found is that we've got a huge percentage of data that's non-coded. It's free text in the same field that the coded text is in. And it turns out a lot of the national indicators only use the coded values. So it's not the fact that that's necessarily wrong, but when it's 40% of it's not coded, you've got to ask yourself, are these national indicators worth anything? Yeah, it's really quite fascinating. I find, yeah, the more you do this sort of work, the more you uncover.
Thanks, guys. Yeah, no, I mean, the qualitative part is really so important, and the contextual part. I mean, there's also other literature that shows other studies that shows that the quality indicators in Australia anyway, so aren't really reflecting the truth at all. So we're confirming it with our own data, which is really great. I can't see any other questions. So perhaps while we got about 10 minutes left. Sorry.
There is one from Karen about whether or not sort of the conversion to the CDM has its own data quality concerns. Is the underlying semantics changed with the mapping?
Mm-hmm. Well, and I think there's actually been quite a few papers written about essentially comparing like the source data to the data once it's been converted to the CDM. And I'll try to look a couple of those up for you, Karen, while we keep talking. But I know that this question is a big one among many folks. A lot of people are concerned about this issue. And I know of at least 2 or 3 papers that have been written showing that there is no data loss and that the data is preserved as it's moved to the OMOP common data model.
Can I jump in? So, I mean, I guess my comment was not so much about data loss or indeed data. Yeah, it's not really about, whether you lose the original data, because I know in OMOP, you point to the original data. So as much as possible, you kind of retain that. It's actually maybe a broader question around the semantics of the common data model itself and whether what the mechanisms are, I suppose, for kind of just checking that, when you pick a particular element of the common data model, that actually the semantics of that really does align correctly with the underlying data semantics. And in particular, maybe part of this is what the mechanisms are for kind of updating the common data model, because if you identify a particular thing that's captured in a data set that doesn't map all that neatly to the common data model, what do you do in that case?
Oh, Karen, I'm so glad you asked that question. So for context, I helped to lead the common data model working group to answer that specific use case you're talking about. Because the common data model is so widely used and it is continuing to be used with lots of different data, we know that it's not going to cover every use case or every type of data or interest that somebody has. So we have this working group to continually allow the model to grow and to change. I like to call it a living model because we do evaluate it constantly. So we have a system for doing that. Right now, we have these working group meetings, and we have, if you see something that is missing, for example, one of our colleagues at Columbia University has been asking for a microbiology table. And so what you will do is you write a proposal saying, I think we need this thing added. We have discussions and we go back and forth to make sure we understand why you think this needs to be added and what the benefits are and all those kinds of things. And as we go through that process, we eventually get to a vote where we ask both the working group and the larger community to vote on such a proposal, and then if it passes, it gets added into a future version of the model itself.
Michael?
And I would answer just a slightly different way, in the sense that clearly something that takes an operational data model that may be hundreds of tables big and converts it down to a data model that's roughly 15 tables, is going to be less, there's going to be sort of a difference between, 400 tables and 15 tables. I mean, one of the things that I think most of the common data models do is, this is just not an OMOP thing, is they retain the original provenance of the data element. They not only the original source value as it existed in the original sort of source data model, but what kind of thing is this? So take a drug. Is it an order? Is it provenance? Is it an order? Is it a prescription? Is it a dispense? What kind of drug sort of fact is this? It keeps the provenance. And that's another way of sort of kind of retaining sort of the semantics that you're asking about, by keeping the original, keeping the provenance, where it came from. Now, clearly, when you move over to the standard side, you're sort of squeezed into the allowed values of the provenance. Our electronic health record, has over 120 different visit types. They're squeezed down to inpatient, outpatient, ED, and ED to inpatient. They're squeezed down into 4. But from an analytic perspective, those are the only 4 that matter. We do not need to know that there was a billing event that, if you look at the 120 kind of visits, most of them are there for administrative purposes of the operational system. They have no clinical meaning. So, some things are well worth getting rid of and missing. They are not relevant. And to worry about, oh my God, I don't have 120 visit types, I've only got 4 to choose from. You got to ask analytically, have you lost anything? And there you have to play the game that Claire was just talking about. If we are missing something analytically relevant, there is a mechanism to go back and say, okay, this is why the CDM's on version 6, and there'll be a version 7 when we realize, oh my goodness, we're not doing vent settings right. Oh my goodness gracious, or microbiology, as you say. We'll go back and we'll capture it. So it's a living thing. Now I see why. I got it.
Something that we're hitting on as a bit of an issue. Teng's looked at the past at mapping primary care data from the same GP systems that we're currently converting in Melbourne. And when we look at a lot of things like the free text, we're having to convert to SNOMED for a lot of stuff that's not coded. So we don't necessarily have complete agreement with what someone else has done in the mappings before. Certainly I see this as being a major problem. And what I'm hoping we can do as one of the activities we're taking forward in Australia through the ARA Transformational Data Collaboration is to try and come up with tools and mechanisms where we can have communities feeding into what these mappings are, rather than them being things that are always hidden, things that are often seen as a very private intellectual property, that companies try to sell. This is something that we're really trying to work on with the Australian Research Data Commons, to not just have somewhere for mappings, but have a system that gives people that flexibility to go in and do things in a community, and try to gradually improve the quality of things over time. Yeah. See what happens there.
Yep. I hope that answers your question. But I guess that raises another question for me. If we have the processes in place in terms of the governance and the provenance, do we also need some form of kind of technical solution, well, more technical solution, in addition to the common data model, as well as the common metadata model? Because I mean, especially Michael, I said is very contextual. It depends on the research question, which is or the intended purpose. So it will be kind of infinite kind of permutations that you have on your data set. So the provenance is important. You should be able to get back to the original data to apply, the different version of the model, or or a different common data model, or whatever. So so I'm just throwing out to the panel.
I got nothing for you, Tang.
Karen, you might have something, Karen. Yeah, muted. Oh.
I'm sorry, my family was actually distracting me. And right when you were asking that question, I was trying to get rid of them, but they kept talking to me, so I didn't actually hear what you just said. I'm sorry.
Sorry, I'm just saying that the protocols and processes you have in place for governance and provenance is important, especially the regular iterative data quality asset checking. But I'm seeing whether we can actually do something by enhancing the current common data model with a common metadata model. But which requires, kind of like most trying to model things, you have to structure things and then provide a taxonomy and a hierarchy and those kind of stuff, which might lose the semantics that you were concerned about.
Some of that, of course, has been done through the UMLS, right? So a lot of what the UMLS is trying to do is organize concepts, and they provide semantic groups, and they provide a kind of semantic backbone, which provides relationships between things. And from my perspective, that's really very valuable information. And so of course, because the common data model in OMOP builds on UMLS concepts, like a lot, there's a lot of interaction relationship between them. I mean, maybe what it means is we need to be doing a better job of remembering that there is a thing called the UMLS, which organizes these things and sits kind of complementary to what's happening in OMOP, and perhaps look at ways to leverage the ontological structures that are already there.
Thank you. Others? Dougie, Claire?
So I think metadata and a metadata model has been on the slate for many years for OHDSI, and I think many people have attempted it, and it has not come to fruition. But it keeps coming up. So it comes up in the CDM working group. I know it's come up in the data quality meetings I have around development of the tool, and that there's always this need for metadata, and no one has really figured out how to do it, at least for OMOP and for OHDSI. And so I think it would be great, and I think we're still working on it, and we definitely need some sort of like interoperable metadata model that works with the OMOP CDM and perhaps other models as well. So I definitely see a need, but I don't have a good answer for you as to how we could get that done.
Sure.
So, I mean, doesn't it depend on what you want to capture in the metadata? Because, I mean, Michael already mentioned the provenance and so on, and that's a kind of there is a model for connecting the transformed data to its original data. So that's a kind of metadata model. What other things do you want to capture in the metadata model?
So I think the problem with one of the attempts that was made a couple years ago is that they tried to capture too much, and that they wanted to capture things like what Michael showed, that graph that he got from Jeff Brown, where like it was run 7 looked good, but run 8 had this like crazy dip. That you would capture that crazy dip in the metadata somewhere, so you have a record that yes, we know this happened, and maybe here's why if we know why. And so that's really where, like a couple of years ago, where metadata was really starting to go. Like, for example, one of my databases has U.S. death information, from a federal file that's held somewhere. But then they lost some of that information, but then they got it back, and so the death data looks all crazy. So we're saying, oh, we can capture that in metadata. But then then it just gets into like this kind of like snowball effect of like everything becomes metadata. And how do you capture it all? I think they ended up with like, and this one attempt, it was something like more tables that was actually than were actually in the full CDM itself, just to capture metadata. And so I just remember going, guys, like it's just not tenable. Like you got to come up with a different solution, and they didn't quite make it there. And so I think that's the first question. I think you're right, Karen, is what do we want to capture? And I think we're still trying to figure that out.
I'm just conscious of the time. It's 5 minutes past the ending time. I don't know, I'm having something with my chat. I can't see all the questions, whatever it is. Can you see any that's there that we should be addressing from anybody? No, okay. Any last final comments from the panel?
Thank you. Thank you for attending. Thank you for having us.
Yeah.
Yes, thank you so much. I'm
Okay.
very happy to be here.
So I think Nicole was thinking, obviously we were wanting an opportunity to go through the sort of poster questions that people might have, and I'm not sure that all of the people doing poster presentations are here. So Nicole's suggesting maybe I can summarize what where we think we're going with OHDSI Australia in the coming year, and then we can maybe turn on all of our screens and welcome everyone to continue the discussion. How does that sound?
Well, there is an activity that is starting up now, right?
Yes.
There is a make fun of everybody else activity coming up.
That's in a half an hour's time, Michael. So we've still got 20 minutes in this session, and then we'll switch back to the live session.
Yeah.
Okay.
All right, okay. So, you're still here, Nicole, you haven't gone it yet. So I guess the other thing is, one of the things, because it's such a broad and evolving area, what I think just when Claire was talking, I was thinking that we maybe should have specific sessions on some of these issues that's been raised here. I think it'd be one way, especially to contribute to the quality assessment, data quality assessment work done in both environments, the more generic and the CDM, in the context of the numerous CDMs around, and so it might be something worth thinking about that it'd be part of the White Bandicoot work, maybe.
Yeah. Yeah, certainly, Tang. I'm really interested in us continuing to develop the tool really to sort of validate it. It's doing the sort of things that you're looking for, I guess, out of it. Make sure it's sort of fit for purpose. Certainly, I can see that being one of the activities which it'd be great to advance in the coming year. And I guess I'm really interested in working closely with Claire and the international community just around what does the dashboard look like? What do you think of the idea of maybe something that's web-based that can act as a collection point for lots of different quality assessments that people can go in and look at that, whether they're from OMOP databases or something else. But what you've got, Claire, is more advanced in how you would present it. So we're just sort of playing around a little bit with how we can do that web presentation. But it's early days for us. So yeah, it'd be good to work together.
Yeah, I guess in terms of sorry, go on, Claire, go on.
I was just going to say, I would love that. I absolutely love the White Bandicoot tool, and I know there are many people that have been asking for that exact thing. I can specifically think of my advisor right now, who's heading up the Eden project, has specifically asked me about source data quality assessment, and I think what you've done is exactly the ticket. And I would love to figure out how we can bring them together, with the DQ dashboard as well. So yeah. I'm very, very excited.
Well, we're very happy to chat about that. Obviously, it's we're really keen to do that. Absolutely.
Yeah. That's an opportunity. I guess with this current the funded project that's going ahead January, could be some opportunities there to apply some resources to both to both Bandicoot and dashboard the OHDSI dashboard work to.
So one thing I would be excited in sort of thinking about is sort of what I see happening here is sort of there is work being done sort of on the source side. There's work being done on the CDM side. You really want to have them side by side. This is getting at Karen's issue about what has happened in sort of the magical transformation from A to B. You really want them to travel together. I'm also interested in this fitness for use for particular use, and I've really been struggling with how do we crowdsource data quality? The renal people are going to drill into the renal functions like nobody else is going to drill into the renal functions. And the liver folks are going to drill into the liver data like nobody else. I mean, when I was doing pediatric terminology mapping, we would deal with the individual subspecialties, and they would go where no one else would ever go in terms of the drill downs. And to be able to think through how do we marry the kind of annotations that you're doing, Dougie? Many of them are manual annotations. They're manual assessments. Who cares how they're computed? Yours are computed in someone's head. Claire's is computed by a template. How do we layer so that the renal guy, when I come in and I'm not a renal guy, but I want to do something with creatinines, I can leverage the fact that the renal guys have squeezed the heck out of the renal functions, and I don't have to deal with it. They've already sort of investigated it far more than me. Layering on all of these different layers so that we can crowdsource our work. I'd really love to see how we could marry what you're doing, what Claire is doing, what other people are doing, and have that ride with the data.
Yeah, I've got some ideas. So certainly, Michael, the thoughts around White Bandicoot are to try and help address that question. Obviously, it can do the calculated things as well as that human thing of putting the annotations in. But what we're thinking here is that every time you've got a new researcher coming along with a project, they're bringing their context with them. And what it means is then they might be picking certain tables and fields of data. And what we're thinking is that just because we've maybe already got some annotation about a data set previously, when that new research question comes in, it's an opportunity to reassess what we've actually recorded against the tables and fields around that researcher's question. So the idea is that it's not static. Every time a researcher comes in, you would again check the particular tables and fields in question. And then with the web-based solution, the idea is that you can give that researcher a specific report because you can pass back to them just the table and fields that are required to answer their question. And they then can see all of the warts or issues or ways the data was originally intended that might impact the ability to actually answer the question. So hopefully that helps. There's no I don't know of any automated way to do it. I think that context changes each time, but certainly being able to just go in and add in that, look at the data set against the context of the question as they come along, and once with our primary care data set, we've now had it's been going a couple of years. We've had 44 different research questions fired at it, I guess. So each time we get another question, it helps us refine that and address the issues of the different context for different areas that the researchers are addressing.
Yeah, back to the fit for purpose principle.
Yeah, so it might be that the answer is no, you can't answer your question in this data set, don't use it.
Yeah, in fact, I mean, one of my heads chaired the ethics committee for the College of GPs here, and one of the provisos we made of kind of condition that, or strong recommendation to data custodians like NPS and so on, is that if it is probably unethical to provide a data set to someone who's applying, to use that data set to answer a research question, it's your obligation to make sure that the data set that you provide to the researchers is actually fit for purpose to answer, which means it needs to be able to answer the research question.
Yeah.
Well, and to that end, I've been thinking a lot about, we've talked a lot about database data quality, and I've been thinking a lot more about, in the larger OHDSI Symposium, or the US OHDSI Symposium, excuse me, earlier this year, they presented the phenotype library. I don't know if you guys have been familiar with this at all, but they've put together this way of defining phenotypes in observational data. And it got me thinking about how data quality is, how the quality maybe of a phenotypes, if you just take one cohort and look at the quality just of that cohort, how does that compare to the larger database? And trying to think about this fitness for use question. So not necessarily just in this structural, can this database answer this question, but should this database answer this question? And we've been trying to think a little bit about how can we pinpoint into that. And I don't know if cohort and phenotype is the right way to go, but that's kind of the avenue we've started to walk down.
So Nicole's letting us know we've got 15 minutes to wrap up. Yeah.
So what else have you got in terms of what's next for OHDSI Australia, Dougie?
Okay, I'll not bother sharing any slides or anything like that. But I think obviously I think in Australia we're really fascinated around these issues of data quality. So I think we certainly, for OHDSI Australia, we really want to be working as part of the international community, and we're excited to work with everyone and see where we can take that. In terms of other aspirations, of course, we've got a huge amount of work now. Things seem to be kicking off in Australia in terms of undertaking common data model conversions. So I think Teng's project is very exciting in terms of being well funded to really look at pinning down the science of these conversions, which is fantastic. We've got an awful lot. So some of the work we've got coming up, we've got mappings happening in Melbourne on our primary care data set. So there's the National Prescribing Services mapping. We're mapping the Victorian Comprehensive Cancer data hub, which contains data from hospitals as well as registries and things like that. And yet we recently got funding to look at starting mapping some of the major hospitals in Melbourne, the Austin, Western Health, and the Peter Mac. And very excitingly, this funding is also to map all of Queensland public health. So we're going to have all of the public health for the whole state of Queensland mapped. So for me, where I want this to go is for this to actually be a demonstration of capability, such that we can actually go to government to try and do this more systematically across the whole country. We've also then discussed today some of the challenges around semantics and so on. So how we can curate and manage not just the ETL, but the knowledge of how original source systems can be converted into a standard, and also the terminologies is really important to us. So for the terminologies, we've called our approach to this, we've called Rosetta. If you're aware of the Rosetta Stone, of course, it's got multiple languages in there. So our thought around this is being able to have a platform that is the Rosetta Stone for us in Australia and Australian researchers, where all sorts of custom mappings can be held. And we're looking to base that on the on a technology called Onto Server, which is used for the National Clinical Terminology Service in Australia, and it's just been adopted in the UK to do such a Rosetta for mappings there. So I think they've got about 3,000 different data sets that they're looking to hold in there. So I'm not wanting to discount the very fine work that happens in OHDSI around the standard terminologies and conversions, but this is about being able to look at very specific Australian problems, I guess, in there, but also make it compatible with what is a national platform that many are already using. So I think there's some of the things that we're looking at, and I think OHDSI, one of the really exciting things about OHDSI Australia for me is the steady stream of the webinars that are going on, where we're able to have fantastic international presenters, and for me, we've been playing around in this space, and it's great to feel that we're coming into in the international community and meeting people like yourself, Michael and Claire. So what will be happening next year? I think a lot more of this networking and driving things forward. And I wonder, Claire, as our president of OHDSI Australia, what are your thoughts on where we're going to go?
I think you meant Nicole, but
sorry, Nicole. You got a new job, Claire.
I thought you had a hit.
I'll take it.
Claire.
I'd love to be with Claire. Yeah, I just want to reiterate what Dougie has said. We have an extremely exciting opportunity here in Australia to really get going with our work in OHDSI. I think things are coming into play. The researchers are coming together, and I'm really excited that we had over 30 participants here today. I've been keeping a track of who's been on and listening. So I think we've got this groundswell happening here now in Australia, and it's really exciting for me to have friends in the community, have friends here in Australia. And I will say that next year we are hoping to actually host the next APAC Symposium here in Australia. We are thinking perhaps around the time of the AMEA conference that is meant to be happening here in Sydney in the middle of next year. I think it's set for June or July. So I was hoping to have a face-to-face OHDSI APAC meeting around that time in Sydney, but we'll wait and see how COVID goes and how things play out with that. But either way, we will meet as a community again next year for APAC and hopefully have a great presence for the Australia chapter in that meeting. I think I'm going to leave it there for now, and I'm going to have to pop off this meeting and head to the live Fun with the Community session, so don't miss it. It's going to be awesome. Pictionary session with the community. Lots of fun. So Dougie, I might leave it up to you in case anybody has any other questions or want to introduce themselves or ask questions of the poster presenters. I think we've got a couple online. So once again, thanks everybody, and thanks for joining us.
Yeah.
Thanks, Nicole.
Yeah, thanks, Nicole.
Karen's
put up.
Thanks, everyone.
Oh, yeah, thanks, Ty. Nice to see you. I see Karen's put up a comment. We've got MedInfo in next year's in Sydney. So that's terrific, actually. Yeah, we will definitely be at MedInfo next year. Yeah, we'll just need to think about that, but we should definitely do that.
COVID permitting.
COVID permitted, yeah, absolutely. So yeah, I mean,
well, what it sounds like to me, though, it sounds like Australia sort of writ large is turning a corner on sort of the idea of broad-based data sharing networks. I mean, I think you, Dougie, opened up with a fairly negative comment about or maybe it was you. No, I think it was you, Doug.
Yeah.
About sort of will we ever get anything out of all of this effort? Will it ever be, irrespective of, whatever that, garbage in, garbage out? But it sounds like you're starting to sort of chip away at some of that resistance and starting to be able to get some fairly large scale pilots going, some significant scale pilots going, where I've known Tang now for maybe 5-7 years, something in that range. Early on, it just didn't feel like the large institutional backers, data owners, were very interested. OntoServer, I think, is a really is that the right name? OntoServer, is that the right name? I mean that's a great example of people being willing to throw their terms into a pot so that they can be sort of brought together and I couldn't imagine that happening 5 to 7 years ago when I first met Tang, that people would be willing to share their data dictionaries and their variables and their labels and their sort of their value sets. So it really does sound like the writ large, the community has kind of turned a corner rather, in some of its resistance. A lot of work to do, but it really feels like new things are possible that weren't before.
Yeah. And, it's not necessarily because obviously there's an awful lot of work and people have been banging their head against brick walls for a number of years, but there's I think the other thing is that as things like OHDSI has matured, our government administrators can now see what's possible.
It's right.
It's
not so abstract. Yeah.
That's right.
Not invented here syndrome and things like that.
That's right.
Well, everybody's also sort of bought into the machine learning, AI, neural net, magic, the whole big data initiative, and you can't do it when you're isolated in small isolated GP practices, and to the degree that you think that those are real game changers, and you want to be in that game, you got to do the stuff that we're talking about,
Yeah, I think things like COVID, as well as focusing everyone's minds, we need the data. So there's been excuses, lots of excuses in the past, and not necessarily wanting to invest or seeing the advantages of investing, but I think certainly perceptions are changing fast.
Was COVID the reason? Was it Queensland, you said, was the public health system that's going to sort of participate in a project?
No, it was that one's more around the vision of a few people that have been talking about this for many years. So I think they've been wanting to do this, and then all of a sudden, because we've now got a national collaboration which is non-competitive, and we're able to do these things for everybody, it's the right environment for this to happen. So yeah.
No, I think what's happened with the ARA is a good, really significant move. It was pretty hard being a lone voice in the wilderness for many years.
I know this.
You will appreciate, but I think it is not just the constant hitting your head against the brick wall, but also the efforts of the international community. The ability of OHDSI to actually start kind of notching up its credibility.
Yeah.
Sorry, I just had a question about the poster session. Is it supposed to be now, or?
Yeah, I think so.
Preetham is here, so I just wanted to check whether it's supposed to be now, or.
Yeah, maybe Preetham would like to share his thoughts on what he's presenting.
Yes, absolutely.
Yeah, okay. Hi, everyone. My name's Preetham. I'm a fourth year medical student at UNSW, and as you may have got from that, I did a little project on the OHDSI kind of data set. So I know what you're talking about today is flying a little bit over my head, because I think it's more around the actual processes of extracting the data set and making sure the data set is essentially clean. But we went that one step further and actually tried to investigate some kind of community-based prevalence patterns of statins in the community, and looked specifically at a high-risk community in Sydney, Australia, southwest Sydney in particular, and looked at the different trends that we saw. So I'm not sure if you've had a go at having a look at the poster, but there's quite a few different interesting trends that we saw there. I think the first thing is that, well, in terms of the prevalence of statins, we saw that there were different decisions being made at a general practitioner level that affected the health of these individuals. So things such as incorporating evidence from new guidelines that have been published and kind of trying to compromise with patients regarding their treatment were certain things that you could actually extract from the trends that you see in the data set itself. And then we went on to compare it to the NHANES, which is a population study from the United States. I'm sure those of you from the United States probably know that study quite well. And we saw that the models that we created to essentially see how well CRP was explained by factors measured in our data set was very similar to the model created in the NHANES. So we actually somewhat validated the use of this kind of data in essentially being used as a gauge for the health of the population itself. And then we went on to a further step and looked actually at the inflammatory hypothesis of atherosclerosis, which suggests that inflammation is involved very heavily in cardiovascular disease. Unfortunately, we didn't find many things in that regard, but I think the findings that we did have in terms of the fact that we could use this data set very well to actually monitor things at a GP scale, and that we could identify particular behavioral trends from essentially this kind of biochemical data and visit data was really quite interesting.
And what's really cool about what you just described was by comparing to NHANES, which is a very expensive survey-based data collection environment, that, I mean, getting a little bit at this cost model that, Tang, you're going to be sort of looking at with the upcoming program. NHANES is incredibly expensive to sort of pull off, because it's a manual randomized survey of people that they have to call up and give questionnaires to. And here you've been able to do it with an electronic data set.
Mm-hmm. Exactly. That's what we saw was really interesting. And I think I also, just out of my own interest, I had a look at what we've done in Australia to maybe replicate the NHANES. And they've actually done a pilot study here where they showed that it wasn't feasible because of that kind of cost barrier that you've mentioned. And that was done all the way back in 2004. And now we have a way of kind of having a proxy measure to get that kind of data.
Yeah.
So it was brilliant that you guys went to NHANES and said, we can match this, because again, now you've got that value proposition. Look, we can do something with these secondary data that we simply could not afford to do any other way. It's a huge, I mean, yes, there's the scientific medical care findings that you have that are important about who is and who is not. And I'm assuming you couldn't do the inflammatory hypotheses because people weren't getting C-reactive protein and those sort of inflammatory markers on a routine basis in general practice. It tends not to be what is measured. But still, irrespective, I mean, you now are able to not only show the science, but show the feasibility of that kind of science.
Of course, yeah.
Yeah. No, I mean, that was, as I said, that was, I mean, as Pritam said, that was done on a small EPBRN data set.
Yeah.
Maybe something can be replicated on Patron, Dougie. That might be useful. And I don't know whether Pritam will be interested.
Well, we'll have to have a look because we're going back into hospital next year. But I think Leung was quite interested in that kind of stuff.
Yeah. Actually, but I noticed Anthony Sanjaya is on this chat group as well. He's doing something in breathlessness, which something that, kind of a larger data set like Patron might be interested in. I mean, MPS is obviously the other one, but,
Yeah, absolutely.
You want to say something, Anthony?
Yes. Hi, thank you for bringing it up. Yes. So we're interested to look at patients with breathlessness as a symptom. So it's not a disease kind of thing, which might complicate things.
Yeah.
But I certainly hope that, if there's a data set that records such a thing, well, that would be useful, certainly.
Yeah.
Yeah.
Yeah, it's a symptom. Yeah.
That's right.
Yeah. Not
We
a
thought the
disease.
primary care data set might be useful, but I think all our data sets actually focus a lot on asthma, not on breathlessness as such,
Mhm.
you know, in the age group that.
Yeah, well, asthma is a recognized disease and it's coded as such. Breathlessness is a symptom.
Yeah, that's right.
Okay. I just want to say, are there other poster presenters still here that want to ask something? So certainly we're being asked to get back into the main.
All right. Okay.
The main meeting, but I really don't want to cut off the poster presenter, so I'm happy to stay on a bit longer and chat.
I think the other presenter was Guy and Tai, so it looks like they, yeah.
That's evidently Dougie, Guy.
Yeah.
Yeah.
Guy Safnat.
Yeah. But actually, you know, Michael and Dougie and and Nicole, I guess if she's still here, is on this project.
Claire.
Yeah. I was thinking of Nicole because she's also on the project that's starting in January next year.
Oh, sorry.
Yeah. One of the things is, you know, the pediatric group is really interested in the continuity of care aspects of this, you know. So I think I might put you, I mean, Valsa, who's the other, you know, co-invest, CI is the professor of pediatric psychiatry, I think, adolescent psychiatry. She's really interested in so that's something that we can actually maybe develop as part of this.
Yeah, absolutely.
Yeah, you have to be careful. I mean, I can only sort of speak from sort of my perspective on the pediatric care. I work in a tertiary pediatric hospital with very little primary care. So almost all of our outcomes truly occurs out in the primary care network, which is separate from us. There are other pediatric facilities in the United States that have a very well integrated system. We're very disconnected. I don't know how you are there. So record linkage is everything. We've got to link across the systems to be able to because those outcomes are happening in the primary care pediatric environments, not in our tertiary care pediatric. So we see nothing. We see nothing of our outcomes using only our data. So somewhere in someone's presentation, there was a record linkage sort of suggestion. If you're going to go down pediatrics, and if you have the same kind of disconnect, I don't know if you do, between the specialist pediatric care and the general pediatric care, then we really need to think carefully about record linkage. I've just started to marry my data quality interest with record linkage. I have a faculty member who's interested in record linkage, and we've now kind of married up together to try to understand how record linkage is both affected by data quality and can record linkage improve data quality. I mean, it works in both directions. In what way does data quality hurt record linkage? In what way can record linkage help data quality? So if you guys want to go down that route, because I know Australia is very aggressive in record linkage activity. You got a lot of stuff going on, especially over on the western side. So maybe there's an angle there.
Yeah, so certainly we did actually present at the International Data Linkages Conference.
I saw that. Yeah.
About a month ago. We're very keen to work in that space. So that's certainly one of the areas I work in as well. And we've got Jim Buttery here is left, who actually works in the Royal Children's Hospital in Melbourne. So Jim's saying we do have the same disconnect in Australian pediatrics, but I'm very keen to work with Jim actually to see if we can get the hospital data there converted to the common data model, so indeed we can link it with our primary care data and other sources. So it'll be fascinating.
No, that'd be really great. In fact, as I said, Valsa, who is the pediatrician in the group, the team, her plan, one of her other project is what she calls what is called a data observatory for the first 2,000 days of life.
2,000 days.
So she started the program. It's called Ethics Approval to recruit 10,000 births from the Southwest Sydney hospital system and start to link the data sets from different data sets, not just and including general practice, to start to follow the cohort. So that'd be something that we linking up with or liaising with Western Australia to do that. But I think it is something that Children's Hospital, if they already not done it or doing it, to be interested in.
Yeah, there is an initiative, Generation Victoria, where they're looking to recruit every birth over the next couple of years, do the similar thing. But look, we're actually really out of time here now, I think. So Jim's put up there, he's mentioning Generation Victoria.
Right.
Karen's put that up as well. I think as the person who's mediating this, I'm going to get into trouble if we don't end it. But it's a shame, because I feel we could talk all afternoon.
Don't you want to go and have some fun, Dougie?
Yeah, so I'd like to just thank everyone here today. I think it's been fantastic. I don't know what time it is where you are, Michael, or what time it is with yourself, Claire, but it's been fantastic.
It's 11 o'clock at Claire's time, yeah.
So thank you so much, and we look forward to seeing and working with you all in the coming months and years. Thank you very much.
Thanks to everybody for staying on too. There's a large number of people down below who stayed on the whole thing.
Yeah. Yeah, and I have to add to that. Thank you for being part of the panel, as participants as well as our audience.
Yeah.
But especially Michael and Claire and Dougie and Jitendra. Thank you very much.
Yeah. Thanks, Aidan. Take care, everyone.
Thanks,
okay, let's go and have some fun.
Yeah, let's have fun.
Yeah.